Natural selection and genetic modification. Alfred Russel Wallace. Alfred Russel Wallace developed theory of speciation and evolution by natural selection. He found that species that don't have the characteristics to survive a change in the environment will die out. He published his findings with Charles Darwin. Process of speciation. 1. Variation happens because of genetic mutations. 2. Alleles which give a survival advantage are selected through natural selection. 3. Populations are isolated by a natural barrier, like a volcano. 4. Different alleles are advantageous in new environment, so they're selected. 5. Overtime selection of different alleles increases genetic variation of two populations. 6. When species can no longer breed together to produce fertile offspring, they are different species. Charles Darwin found theory of evolution, which was published in On the Origin of Species. Theory of evolution. Variation happens because of mutations in DNA. Organisms that have characteristics best suited for the environment can survive, so they can reproduce and breed successfully, and this is called the survival of the fittest. These characteristics are passed to the next generation, and the characteristic is then passed from one generation to the other, which allows for all organisms to have the advantageous characteristics. Controversies for these theories. 1. Contradicted God as the creator of all things. 2. Few studies done, so not much information to back up the theory. 3. Mechanism of variation and inheritance were not known at the time. Emerging resistance. Bacteria are resistant when antibiotics that used to kill them no longer do this. Bacteria reproducent fast. Mutations in reproduction can result in antibiotic resistances, so create a new strain. Being exposed to an antibiotic creates a selection pressure, so those who are antibiotic resistant don't die, and the ones without antibiotic resistance die. Antibiotic resistance reproduces and passes down advantageous gene to offspring, so resistant bacteria being present shows how Darwin theory was right. Populations of antibiotic-resistant bacteria increases and bacteria diseases spread faster, because people are not immune to new resistant bacteria and no treatment for it. Can spread in hospitals as doctors move from one patient to the next. Fossil evidence for human evolution. Fossils show how evolution in humans happened over a long time because we use carbon dating and other techniques to find when a fossil was made, so we know how a species developed over time. Examples. 1. Artie was the oldest human ancestor who lived 4.4 million years ago. Her fossil's bones showed she was somewhat human, but also ape, so Artie was phenotypically somewhere in between a human and ape. This is evidence of natural selection and evolution over time. Bone structure tells us that Artie's feet were different to a chimpanzee, which means that the two evolved separately rather than together. 2. Lucy lived 3.2 million years ago. Her bones show that she walked upright and had human-like position. But Lucy had a small chimp-like skull and brain, so shows she was something in between apes and humans. 3. Fossils of Lewis and Mary Leakey found, which help support the theory of natural selection, especially an early fossil, which has remnants of stone tools and Homo habilis, which now is one of the most important early human species. Dating stone tools and anatomical evidence for evolution. Use stone tools as evidence for human evolution. It's clear how stone tools have become more complex as our brains evolved in complexity. Early Stone Age was called Homo habilis, which were 1.5 million years ago. They used basic pebble tools created by smashing rocks together. These tools had simple uses like cracking nuts. Late Stone Age tools were used by Homo neanderthalensis 40,000 years ago and modern Homo sapiens. These more advanced species used pointed arrowheads, spears, and hooks. This allowed more advanced tasks to be done like catching fishes. Data tools using two methods. 1. Radiometric carbon dating, which looks at the natural radioactive decay of an isotope of carbon-14. Using this, we can find how long ago an organism lived. If a living material was found with a tool like piece of wood or fur, we can date this to find age of a rock. 2. Stratifying rock layers is when each layer of sediment is when a rock was found is a useful tool for archaeologists. Each altar of sediment and everything in it needs to be formed at the same time. So, data once living fossils in this layer and use this to estimate when the tool was formed. Look at how anatomical features developed over time using fossils gives important evidence for evolution. Pentadactyl limb is a limb with 5 digits, seen in a lot of organisms showing that they come from a common ancestor and that each branched off at some point in evolution. This could be because of different selection pressures in different environments. Human hands have 5 digits, but bats, cats, horses, and birds also have this pattern in their limbs. But that doesn't mean that we evolved directly from animals, but humans are distantly related to them by common ancestors. 5 kingdoms and 3 domains. Classify organisms to find genetic relationships between species and organisms. Do this by using 5 kingdoms and new 3-domain systems. 5 kingdoms are animals, plants, fungi, prokaryotes, and protists. Each kingdom then divides again into phylum, class, genus, order, and species, and these can be different for each organism. Example is humans who are Homo sapiens, and our kingdom would be animal. Phylum is Chordata, class is Mammalia, and order is Primate. Binomial naming system is genus name followed by species name, and an example is Homo sapiens, where Homo is the genus and sapiens is the species. 3-domain system created because of improvements of microscopes and increased knowledge of biochemistry found that some species are more distantly related than first thought. Carl Woese added 3 groups called domains above kingdoms. Group 1 was Archaea, which is bacteria that live in extreme environments like hot springs. Group 2 was bacteria, which are real bacteria but have similar features to Archaea. Group 3 is eukaryote, which are organisms who have nucleus enclosed in membranes, and this includes kingdoms protists, fungi, plants, and animals. Selective breeding. Selective breeding is when humans choose organisms to breed to male an offspring with a certain desirable characteristics like animal with more meat, plants with disease-resistant or big flowers. Happened for many years because animals were domesticated and plants were grown for food. Steps. 1. Parents with desirable characteristics are selected. 2. They are bred together. 3. From the offsprings, those with desirable characteristics are bred together. 4. The process repeats until all offspring have the desired characteristics. The problem with this is it could lead to interbreeding because breeding animals with similar desirable characteristics means you might breed close related individuals, which will mean that the gene pool is reduced because the number of different alleles is reduced because they mostly have the same alleles. So, if environment changes or there's a new disease, then the species become extinct because they all have the same genetic makeup. This is important in selective breeding of plants because one disease could spread rapidly and destroy the entire population of plants. Another problem is that small gene pool means there's a greater chance of genetic defects present in offspring because recessive characteristics are more likely to be present. So there's a higher frequency of genetic conditions than normal. Genetic engineering. Genetic engineering modifies the genome of organisms by introducing a gene from another organism to give desired characteristic. Plant cells have been engineered for disease resistance or to have larger fruits. Bacteria cells have been engineered to produce substances useful to humans like human insulin to treat diabetes. Stages of genetic engineering. 1. Genes from chromosome are cut out using restriction enzymes. 2. Same restriction enzymes are used to cut vector like virus or bacterial plasmid into which genes will be placed. 3. Ligase enzyme used to attach sticky ends of gene and vector together to produce recombinant gene product. Vector placed in another organism at early stage in development, so desired gene moves into its cells and cause organisms to grow with desired characteristics. In plants, the vector is put into meristematic cells, which can produce identical copies of modified plant. Implications of genetic engineering and selective breeding. Benefits of genetic engineering. Useful in medicine to mass produce certain hormones in microorganisms like bacteria and fungi. In agriculture, it improves yield by improving growth rates. Introducing modifications that allow crops to grow in different conditions like hotter or drier climates, and introducing modifications that allow plants to make their own pesticides or herbicides. Crops with extra vitamins can be made in areas where they're difficult to get. Greater yield helps solve world hunger, which becomes an increasingly bigger issue because of population growth. Risks of genetic engineering. Genetically modified crops have effect on wildflowers and insects. Genetically modified crops are infertile, and these genes could spread into wild plants, sow infertility in other species which affect entire environment. Growing with herbicides and pesticides can kill insects and other plants which reduce biodiversity. People are worried that we don't completely understand the effect of genetically modified crops on human health. Genetic engineering in agriculture leads to genetic engineering in humans. This results in people using technology to have designer babies. Create selection pressure, which could lead to increased resistance in other species, creating super weeds and pests. Benefits of selective breeding. Possible to greatly increase yield of particular crop by selectively breeding only individuals that produce higher quality or larger mass of food. Individual plants or animals can be bred to be resistant to particular disease, which could increase crop yield. Drawbacks of selective breeding. Selecting for advantageous characteristics can sometimes cause severe health problems in offsprings. Lack of genetic variation. Even though bred population being able to have resistance to disease, if one of them has susceptibility to different diseases, then they all do, and the entire population could be wiped out because of this. There are ethical issues with selective breeding. People consider it unethical to selectively breed for characteristics wanted by humans if it means that the offspring will suffer or have a reduced quality of life because of this. Thank you for watching. Please like, comment, and subscribe.